This is part of the ICGB seminar series, and it is also part of the BNOC Africa program. BNOC is an ICGB program that works towards the development of bioinoculants for agriculture and food security in Africa, and it is funded by the Ministry of Foreign Affairs and International Cooperation. I am Elena Benedetti, for who is not from ICGB, the head of the Fundraising and Technology Transfer Office, and here with me is Fernando Cantao, that manages the BNOC project. This seminar will be delivered by Dr. Fernando Hercos Valicente. He's one of Brazil's leading entomologists. He has an international reputation in his field, and he earned his degree in agronomy from the Federal University of Viçosa in Brazil, and both his master's and PhD in entomology and molecular genetics from Purdue University. Dr. Valicente's research is centered on biological control and with a particular emphasis on the development of Bacillus thuringiensis and baculovirus-based biopesticides. And in addition to his scientific work, Dr. Valicente is also a key advisor in high-level committees and organizations. These include the National Biosafety Technical Commission, the Agricultural Biotechnology Stewardship Technical Committee Non-Target Organism Working Group of ELSI in Washington, D.C., and committees of the FAO and UN FAO geared toward the control of fall armyworm. So thank you, Dr. Valicente, and I leave it to you for this interesting seminar.
Thanks a lot for the invitation and to present the seminar about the biological control in Brazil and the use of bioinputs. And I would like to thank the ICGB for this opportunity to show what Brazil has done in the last 40 years. I'll try to explain and try to make it clear for the Brazilian agricultural landscape nowadays. So I work for Embrapa, that it's a Brazilian agricultural research corporation. It's present in every state of the country. So in the south, you may have like research centers for temperate fruits, and southeast where we are corn and sorghum. We have rice and beans research center. We are 43 research units in Brazil, and Embrapa Maize and Sorghum is in Sete Lagoas, Minas Gerais, and we are 249 employees and 73 researchers regarding corn and sorghum cultures. We are located in the central part, the southeast. Minas Gerais state is in the southeast of Brazil, in the border of São Paulo and Rio de Janeiro. Here is an aerial view of Embrapa Maize and Sorghum and a picture of the city of Sete Lagoas. Trying to understand the Brazilian agricultural landscape. We are a tropical country with 3 crops a year, and in many regions. But this permits the insect pests attacking these crops all year round, because you create a green bridge from crop to crop in most of these regions, and they can survive, adapt, and reproduce. Of course, we provide food, but at the same time, we have a problem: insect pests all over. So when you travel, you can see the same crops or different crops all year round. This is the scenery what we see in Brazil. So, and we can have, you may see cotton, sugarcane, maize, soybeans, beans, rice, and it depends on the region. It may vary, but that's what we see. I think that we not increase the area, expanding the area. We are expanding the productivity, the production per hectare. We have now a major insect pest that we work, and it attacks maize, soybeans, cotton, rice, grass, and we have Spodoptera frugiperda, Spodoptera eridania, cosmioides, and soybean looper, that it used to be a secondary pest. Now it's a major pest of cotton and soybeans. And Fall armyworm attacks more than 300 crops, from peanuts to corn, sorghum, sugarcane, everything. So a brief history of the biological control program at Embrapa Maize and Sorghum. I started very early in the 1984, and from 1984 to 89, we surveyed more than 14,000 larvae in different regions of Minas Gerais state, and 1992-93 from Paraná state in the south of Brazil. We always did some kind of scouting to try to track parasitoids and pathogens, baculovirus. And so we started that very early. And from 1992 to 98, we started our BT collection, collecting soils all over. So this is an old program, but in the beginning, we just survey and try to maintain some of these natural enemies alive. So if you work with research, you have to do sampling, scouting to do your collections. So as I said, we surveyed baculovirus since 1984, and we still do have this collection. And Bacillus thuringiensis since 1992. So it has been 33 years that we have collected soil, grain dust, dead insects, spider webs, and we can get BT from all over. So today, the Embrapa Maize and Sorghum does have a microorganism collection from 18,000 isolates for research and biological products to be put in the market. So our major goals are control pests, diseases, water stress, and plant growth. I'm going to show you in the end some of the products that we have developed and put in the market. But these are the four major goals of our microorganism collection. We have more than, from these 11,000 isolates, we have more than 200 baculovirus for fall armyworm, soybean looper, cotton bollworm, and more than 4,500 BT strains that usually people contact us for caterpillars, sucking bugs, ants, coleopterans, dipterans, nematodes, and human cancer cells. Yes, BT does control human cancer cells as well as human worms. So just when bioproducts began to be accepted by farmers. As I said, I started this like 40 years ago, but biological products had greater acceptance after the detection of the cotton bollworm Helicoverpa armigera in Brazil in 2012, 2013. This was the turning point in Brazil. So Helicoverpa armigera at that time caused losses in the billions, and Ministry of Agriculture had to approve materials to control this insect because we didn't have any. So before they imported baculovirus, I just went to the field and collected more than 3,000 larvae and observed them in the lab. And we got, for example, 8 baculovirus isolates. And I'll show you in the middle of this talk. So why bioinputs worked in Brazil? I was thinking about that because in the early beginning, we just had the isolates, the collections, characterizations, lab bioassays. And we talked to the researchers and yeah, we have this, we have that. In the lab, it can reproduce like this and that. After some time, we had the middle of the process, small scale production, culture media for specific for BT, not for baculovirus. And even for people who worked with fungi, it worked in the same way. Some field trials because we could produce a small amount and try them in the field. And after that, we get the turning point in the middle part of with the formulations. I'll talk a little bit about formulations, but formulations are the key for any company, and it's a secret. So you have to maintain that live organism is stable, viable. So this is the turning point of the middle part, and we got the end, the commercial product. So we got the commercial production and the shelf life. And the private companies put those products in the market. So I think that in the beginning, we didn't have this boom of the bioproducts or bioinputs because we just had the beginning of the process. Now we have the whole package to provide to anyone, at least with the basics for each microorganism. So if you're going to work with insect pathogens, you have to start with characterization, besides like collection, surveying, molecular characterization, morphological characterization, general aspects of the insect pathogen, and must know the pathogen. You're going to know your pathogen. It's amazing, but you know one by one. And after that, you have the field technology for the crop that you work, because soybean is different than corn, it's different than rice. So you have some points to adjust, make some adjustments like nozzles, water volume, the mechanics of application, the nozzles, everything is really important in this field technology. And the production, this is for the industry. So you have small scale and to large scale production. Bacteria and fungi, you have to test media and the conditions, because some strains are very specific regarding to some nutrients. And baculovirus, you have to test temperature of incubation, age of the insect, and concentration. This is the 3 point for the baculovirus. So biological control, some people may wonder why they think that is the salvation, that it's gonna a life savior. Yeah, it is just the tip of the iceberg with these biological control agents. But the most important is the timing. I realize that. The timing is really important when you have a crop in the field, because it depends on the target insect. For example, if you are talking about foreign armyworm, are you talking about maize, soybean, cotton, what crop? Because it attacks differently for every crop. Soybean looper, are you talking about soybean or cotton? It's different. Cotton bollworm, it's the same. And it depends on the region. If it's Brazil, in Brazil, which region, because they plant the crops at different times, in US, Africa, or Asia, New Zealand, Australia. So I just went to New Zealand because we have a project with baculovirus, because fall armyworm just got there. So they're going to start using bioproducts instead of chemicals. It's much easier. That's an advice to everyone who is listening. That's don't start with chemicals. You have everything nowadays for fall armyworm, soybean looper, for biological management and control of these insect pests. Because if you start with chemicals, sometimes you can't find the point to come back and start biologicals. Now Brazil has started with biologicals, and they see the advantages of this biological control. But remember, timing is the most important for most pests. And for most pests, you cannot skip the first spray, because you can kill them when they are really young, really small larvae. Otherwise, you're going to have overlapping sizes of these pests. And most of these biological agents, they work when the larvae are really small. When they are really small, the pH, the intestine, the gut pH is really high. So that's when they work better. So a successful application of these biological products, as I said, is the timing. So usually you do that after 4 p.m. because of the UV light. So every formulation, we try to find a UV protectant that is really effective. Spray between 100 and 120 liters per hectare for those pests that we are talking, not for coffee, that it's different. They are trees. So, And the pH of water is really important, between 6 and 7. Baculovirus and Bt must be ingested. They don't act as a contact, like a chemical pesticide. So the larvae has to eat a little bit to get infected. Spray 1 week after plant emergency. Why? When this kind of fall armyworm, for example, occurs in corn and you have a hot climate, 1 week after the plants emerged, they are there. You may not see them, but you have to respect that the cycle of a fall armyworm in the field, it's 30 days, like 15 to 17 days, like a caterpillar. So it's too short. So in 3 days, they just almost double the size until they pupate. So be careful with the timing and survey that in the field. So this is the fall armyworm on grape on your left. And on the right, it's on corn. And we sprayed when they were in the eggs. We spray the egg masses. So the first thing that they do is they come out and they eat the corn, the eggs, and they are infected. So the pH is really high. They die without causing any damage. And this in the leaves on the grape are really amazing because you can see that they even left the place of the oviposition. Biological control with baculovirus. These are typical symptoms. They are of the caterpillar with infected with baculovirus. This is a baculovirus with an electron microscope. And this is a fall armyworm. That's you have an Y inverted in the forehead. So at Embrapa, maize and sorghum, specifically, we have a baculovirus collection for soybean looper, for fall armyworm, for Helicoverpa armigera. For baculovirus, for fall armyworm, we have about 22 isolates, and isolate number 6 and 19 are the most commercialized with private companies. And this is the only one that we have that does not cause the disruption of the integument immediately after death. The common type, isolate 19, they cause the disruption of the integument immediately after death. So this is the most commercialized because it's easy to harvest and reduce lab labor, and no baculovirus are lost. And I'll show you in the end some of the products and the places where they are commercialized. This is the electron microscope of a baculovirus, where you can see the pulse of the mouth parts, and you can see the whole cell infected, because it's a nuclear polytrosis virus. It reproduces in the nucleus. So you can see here that there's no nucleus anymore, but you can see some mitochondria, and you can see the baculovirus. And here a big picture of the baculovirus, and it's really easy because you can count them in the same way that you can count your red blood cells, Neubauer's chamber. So that's how we used to do the concentration. Baculovirus for soybean looper, we found about 50 of these at Embrapa. We have that in our collection, and isolate number 15 is the most effective. I don't have time here to show the specific results, how we chose isolate number 15, but with baculovirus, we can do many things. We can do polyhedra per caterpillar, total polyhedra produced for 1 liter for you to do your concentration in the field. You have many with different temperatures. I can show you for those who are interested after later via email. Baculovirus for Helicoverpa armigera. It's important for many crops, like fall armyworm, and Helicoverpa armigera and Zea are really common in vegetables like tomatoes, and they can be produced in the lab easily too. We did some, as I said, I collected this baculovirus, 2 to 3,000 larvae when they infested, they came to Brazil. And we did the identification because I said how we got baculovirus if we didn't have baculovirus approved at that time. So in the identification, we used two primers, LEF8 locus and LEF9, and they were the same. So Gemstar was the one that later came to the market. So our strains are close to Australian strain, Helicoverpa H-year NPV for those from BR1 to BR4, and the Gemstar is HZ NPV. So it was collected in Helicoverpa here and adapted. So ours is kind of a different original, close to the Australian genome. It's different. So our baculovirus collection, our isolates were characterized and some were sequenced. I can provide for example, that strain that does not cause the disruption of the integument, it was proved that they have the two genes are responsible for the disruption of this integument, chitinase and catepsin, and both were sequenced, and they found a mutation in the chitinase gene, lacking an adenine in the position 123, so it causes a stop codon immediately after. So it's a truncated protein, so it does not cause the disruption of the integument. So all of them were and are tested against different hosts, and different formulations develop like liquid and powder. And we test that at least twice a year in the field. So formulations, as I said, it's strategic to any company. We do that for baculovirus and BT. And we have, for example, baculovirus at different formulations. We have that for shelf life. And we have powder, we have liquid, we have oil emulsions. And this we do for the shelf life, we test that at time 0, 4, 8, and 12 months after they do the formulation. And this also helps the companies to register the product. The company that signs an agreement with Embrapa, they have to have their own formulation, can provide the basics, but they have to test their own formulation. But we can provide some insights with our formulation because the isolate is the same. So formulation, it's an industrial secret. It generates patents and always in evolution. It never stops. This patent is really important maybe for Embrapa, but for private companies to have this industrial secret of what you add. But we are always in evolution, like, for example, always find some UV protectants. This is the hardest part to maintain those formulations stable for more than a year with some UV protectants, because you may have UV A, B, and C. And so you can provide the protection for all of these three types of UV. So we do field trials at least twice a year. So we do for smallhold farmers with manually doing the spraying, and we do that using tractors and trying to do the best for all types of farmers, smallhold, medium-hold farmers, and everyone. We have the results, and they are really interesting because we try our isolates in different formulations and for commercial formulations from our isolates provided from private companies to see if there is a difference during the production systems. So one thing that it should be clear here is the biologicals and the chemicals. As I said, the biologicals for BT and baculovirus, they are oral in the ingestion, and chemicals mostly are contact. Biologicals, they can be mixed up with the chemicals. Maybe we don't like much the idea, but it can be. We do have tables of compatibility, and and they are compatible with most chemicals. The problem is that some people are mixing more than two products. Maybe in Brazil, they do that with 7, 8, 9 products to just do one round of spray in the field with herbicides, chemicals, and that stuff. So at that point, we don't know what's going on in the tank, in that mixture. If you get, for example, some chemical pesticide and baculovirus, either or BT, it's okay. For most cases, we have a table for that. But when you start adding 3, 4, 5, 6 chemicals, we don't know even the pH of that reaction. We have no idea what's going on in there. So just be careful. Biological control with Bacillus thuringiensis. Let's try to get a little bit with this bacterium that it's really much used all over the world. And you have a spore in the crystal. And we have, as I said, a BT collection. We collected more than 2,000 samples of soil grain dust all over Brazil. And we have then more 4,500, 4,600 isolates. And these are they are kept in two types of freezers at -20 and -80 in different locations for security reasons. And we have also a soil collection at Embrapa, Maize and Sorghum, and we have labeled, and you can see where this soil came from different regions. So many times we want to do something for stress, water stress. So you have an idea from what type of soil you may collect, and you may get 1 gram and try to do your work to select this bacterium to start your work. This is 33 years old, this soil collection, and it still works. So in our lab, we do characterization of Bacillus thuringiensis. We do AFLPs, diversity, plasmid patterns. We do repetitive sequences like microsatellites, ERIC, weapon box. ERIC is the most used. But the most important is the toxic proteins, cry, VIP, and CIT proteins. And people do whatever they do, what they want for the thesis, but what I want is the kill the target insect. That's the major point of our lab. So basically students, they do the toxic proteins and do some ERIC to get some difference. We do plasmid patterns to check, because most of these cry genes are in the megaplasmids, and sometimes it's not many people work with plasmids. It's not easy. We had to do lots of work to adjust the medium to get a really good gel to get this identification. This is just a basic AFLP where you use the combination of colors at the same time, trying to get BT from dry regions, from soil. You get, for example, fertility differences in soil, so you can get BT from these regions. But we didn't get like any good marker. It's so diverse that it's really confusing to try to work. Some of molecular characterization, this is basic for some PCR, but it helps a lot when we have to do some beta exotoxins, some toxins that you may not have in the fermentation systems. So we still use PCR for those, and we can use PCR for these genes, some basic genes, just to make sure that you can find whatever you are looking for. We do protein profile, that it's really important for you to know the size of your protein. And this is quite simple that we do like for most of our isolates that we provide to the companies. And it's a way for you to have a fingerprinting of your strains. So protein profile is itself used to get this fingerprint. For example, for most cry-1 toxins, we have a toxic fragment of 65 to 70 kilodaltons. It helps a lot when you see that in the acrylamide gel. We also try to do some, we ask, we have a sequencing. We don't do that anymore, just some parts. So some companies do that, and we have the interpretation of our strains. For example, in the second row here, we have the number of the strain 474, in yellow 697, pink like 702, and so on. And the other strain 1074. Here in the first row, we have the genes that we found. We have the identity, the length, the mismatch, the gap, and we try to interpret those to use this for biological products and even to provide genes for transgenic plants for some private companies. So we tested our isolates against some specific insect pests. Yes, we tested 4,500 BT strains against fall armyworm. In the beginning, it was, and it is one of the most important pests in Brazil. The best isolates were negotiated, Embrapa did, with private companies for large-scale research and development. Private companies signed a contract with Embrapa, developed their own formulation, registered, and placed it on the market. We provide insights with our formulations, IPM, and biological control field trials, and monthly survey of our armyworm. We also provide data. So Embrapa does and private companies, if someone is wondering how, companies fill out a form with specific objective, like we want BT or a baculovirus for fall armyworm, or a BT for soybean looper and cotton bollworm, and an NDA signed between Embrapa and the private company, and I do the plan of work, or someone else if the interest is like stress or plant growth, and the plan of work is prepared, and a contract is signed by both parties, and the project begins. So what we do the isolation, identification, and bioassays, and we do that with a pilot, a small reactor like 4 liters, and after that, we pass that to the private companies, as we said. This is a private company who does have an agreement with Embrapa. And so these large-scale fermentation systems are like that in Brazil nowadays. This is Balagro company, close to Campinas in São Paulo state. This is Symbiose in Rio Grande do Sul. That is the largest fermentation capacity in Brazil. So it's extremely south of Brazil, and they open another one in Paraná state. And so we have an agreement for 3 baculovirus and 1 BT. They are gonna get the registration till the end of the year. This is Balagro, as I said. They produce 2 of our strains. And Biotrop is a really nice company, a big one in south of Brazil, also in Paraná, Curitiba. And they have a huge capacity of fermentation, but they do more than Bacillus thuringiensis. I want to show the Bom Futuro group. They have 22 farms like this, and they are powerful. And they built a biofactory in one of the farms. They invested $6 million, like 35 million reais. And I went there, and it's amazing. They have all the top-notch equipments and development to try to provide bioinputs for not only BT, but everything for the 660,000 hectares in different farms in Mato Grosso state in Brazil. And that's amazing how they work. They have a plan, and they have nice facilities, really huge facilities that works very well. For baculovirus, large-scale production systems, we have biofactories with controlled humidity, temperature, light and dark periods. This is the latest that was it's an agreement with Embrapa Maize and Sorghum and biofactory in Mato Grosso, and I heard that they invested like $3 million, yeah, in this biofactory. This is a joint program with a cooperative, an institute, and Embrapa. Okay, and this is the biofactory, and this is one of the halls that they have these blue doors, and just showing that the blue door inside you can see these shelves. In each of the blue doors, they have at least 2 million insects. It's a huge production. So for you guys to have an idea, some companies that were working with 500 kilos of dead larvae contaminated with baculovirus, now they are working with 4 tons to 5 tons of dead insects. They are reducing baculovirus at this rhythm, so it's increasing a lot in the last few years. I'm going to try to show some biological products, some bioinputs developed by Embrapa Maize and Sorghum. I want to make sure that I don't, and the researchers of Embrapa don't get any profit. Embrapa does get royalties, but we don't. So this is not an advertisement. I just want to show that we work and try to provide to farmers with bioinputs to try to help the environment in the Brazilian agricultural landscape. So this was developed in Embrapa Maize and Sorghum. It's the latest one called Hydratus, that it's an innovative bioproduct based on a Bacillus subtilis to increase plant resilience to drought. So Dr. Eliana Gomes and the team just worked with that. They got the soil from that BT, the soil collection, and they have many bacillus, different bacillus that they got. She sent me the slides. It's kind of difficult for me to show something that I don't work directly, but they do all the work that to prove that increase resilience to drought. So this is from a bioma company. Hydratus is already in the market. So also we have efficacious control, that is a biofungicide based on a Penibacillus otoi, and Dr. Rodrigo is the main researcher and the team, and the management of soil pathogens. And this was released also this year, and it's a huge step in the control of the management of soil pathogens. So the Embrapa, the team also, Rodrigo Veras, did a really nice job working with this Penibacillus. Biomafos, Dr. Cristiane Paiva worked for 19 years, and she does have like 6 million hectares sprayed with this Bacillus subtilis, 2 strains, and Bacillus megatherium to solubilize or make phosphor available, the phosphate element, and improve the root system of plants. Soil health, low carbon emissions, low cost, low water pollution, or reduction of synthetic phosphate fertilizer. I know that most of these Brazilian companies also did. For example, Bioma Foz was released by Bioma, and they already have an agreement with Corteva. I'm going to show another bioproduct that they have an agreement with FMC International. So most of these Brazilian companies of bioinputs, they have done agreements with private companies to release this all over the world. So this has been done with Dr. Cristiane Paiva and group. And we also did transgenic maize. So that was the first Brazilian maize with Cry1 gene from our collection, effective against fall armyworm. I published this paper in 2010 in a biological control journal. And in 2014-15, we started this project, and the coordination is Dr. Newton. And we got in 2-3 years ago the approval by the technical commission, the National Technical Commission of this transgenic maize for fall armyworm, and developed with Helix, that it's a Brazilian private company, and it's already in the market. So I think that Embrapa Maize and Sorghum has done a nice job trying to use bioinputs and the genes for many purposes. Okay, so now I'll try to show, just going to the end, the biological products that developed by biological control lab that I try to coordinate here at Embrapa Maize and Sorghum. So we have a sera that it was developed by Balagro company that I showed some pictures, and we joined two Brazilian two strains from our collection, and we can control armyworm, soybean looper, and many other pests, but it was released and registered for only two strains. And it also has another registration here. And this Acera, it's a really important biological pesticide because we produce one strain, get it stable formulation, the second strain is stable formulation, and we join in different proportions. It's already in the market, and this company signed an agreement with FMC International. Crystal is another biological pesticide that it was developed with Farroupilha at the beginning. Now it's Laliman, that it's the Canadian company, and it is in the market, in the Brazilian market. And we have three products close to registration. One by the company called Forbio, and the other one from Bayes Institute called BT3, and then another one from Symbiosis, that it will be for fall armyworm. So we have 2 and 3 on the way to be registered till the end of the year. Baculovirus for fall armyworm. So the first baculovirus for fall armyworm was Cartuchovich, and generated the clone that it's a baculonat that bought this company. This is commercial. I just hear that over the news. So they have this clone, baculonat SF. Another company, Promip, has a baculonip SF. This is really important because most people just commercialize the isolate that does not disrupt the integument. And baculonip does the isolate 19 that causes the disruption of the integument. And they have all these registrations to put on the market. So they have the destroyer, that it's isolate number 6. Giromix, that's the huge biofactory that I showed in Mato Grosso, together with Institute and Embrapa and a cooperative. That's isolate number 6 too. And this is interesting because we have Andermatt, the Swiss company. They negotiated with Embrapa isolate number 6 for Brazil, Spodoptera, because it's a Swiss company with the bases in Brazil, but they produce baculovirus in Canada. And they have the isolate number 19 for Africa and Asia. And it's a liquid formulation. We have Symbiosis, they have a Vir Control series, and they have a clone, Laffy Protection for fall armyworm. They have soybean looper, and they have a clone that it's looper. It's a Vir Control and looper. And we have baculovirus for Helicoverpa armigera. Symbiosis, they have no clone. And one thing that I want to show for this, showing this table that we generated, and all what we have seen now is that we can manage these pests even in a tropical country like Brazil and many countries like in Africa, and we can do like hot parts in Australia. You can do that only using biological pesticides. So my advice is don't start with chemicals. Start with biological pesticides and try to do the management. It's easier because if you start with chemicals, sometimes you cannot come back. So you can do that with biological pesticides. So this is a table of we have produced in our lab with the isolates and the places that it's been available. And thank you very much for your attention. Thanks.